This is Dr. Jack Martin, Chief Medical Officer of PeriRx. I would like to take this opportunity to review the scientific background for our Salivary Diagnostics platform. The National Institutes of Health has invested heavily in salivary diagnostics in the last decade. The NIH vision was by 2013 to determine the efficacy of using salivary diagnostics to monitor health and diagnose at least one systemic disease. A salivary knowledge base exists in this website supported by UCLA. It describes the salivary biomarker diagnostic alphabet. Included in this is the transcriptome and proteome. These have been well characterized by NIH-supported investigations. These studies demonstrate that abundant messenger RNAs and proteins exist in cell-free saliva. The salivary glands have a rich vascular supply. Tumors are known to produce mediators. This includes hormones, cytokines, and lymphokines that can modulate the activities and gene expression patterns of distal organs, including the salivary glands, through the vasculature. In a series of experiments conducted at UCLA, non-small cell lung cancer and melanoma tumor cells were injected into the abdominal cavity of mice. As these tumors grew, there was a change in the salivary transcriptome and proteome. In summary, melanoma and non-small cell lung cancer mouse models demonstrate disease-specific alterations in the salivary transcriptome and proteome in the presence of malignancy. The three major human salivary glands are depicted in this slide. The previous experiments demonstrate that animal models support the conclusion that upon systemic disease development, significant disease-specific changes can occur in the salivary biomarker profile and thus set the stage for human investigation. This study conducted at UCLA demonstrated that a combination of 4 messenger RNAs could be used to distinguish patients with oral squamous cell cancer from normal controls with a very robust area under the ROC curve of 0.95 with corresponding excellent sensitivity and specificity. The group at UCLA demonstrated that the salivary transcriptome performed better than measuring the same messenger RNAs in blood for distinguishing patients with oral squamous cell cancer from controls. Salivary biomarkers were used to distinguish patients with oral squamous cell cancer from normal controls in additional populations in Serbia and in India. Excellent areas under the ROC curve in these two investigations highlight the ability to use these biomarkers in multi-ethnic populations. The National Cancer Institute Early Detection and Research Network validated these salivary biomarkers independently in a study that involved 395 subjects. When comparing the results obtained in the National Cancer Institute Early Detection and Research Network Bioreference Laboratory and the laboratory at UCLA, you can see that there was excellent correlation between these two laboratories. In conclusion, over 10 years of NIH-supported investigations have established salivary biomarkers as a viable modality to monitor systemic disease non-invasively. Oral squamous cell cancer salivary markers have been studied in over 600 patients worldwide and have been validated by the National Cancer Institute Early Detection and Research Network. There is a very solid basis for the presently ongoing regulatory approval trial for the use of salivary biomarkers in oral squamous cell cancer detection. These prior investigations also provide a firm background for the presently ongoing investigations related to salivary biomarkers for other malignancies and metabolic diseases. Thank you very much for your attention.